Hello everyone, I am Shanmukha. Today I will be presenting on the subretinal implants. The presentation is broadly divided into 4 categories, where we will be beginning our discussion on the visual prosthesis, then later moving on to the anatomy of human eye, wherein we'll be dealing with certain diseases and disorders which are affecting the normal vision, and then we'll also discuss the role of artificial vision implants in restoring the human vision. Then the third section deals about subretinal implants, wherein we'll be talking about the architecture of the subretinal implants and how subjects who were implanted with these, how it is aiding them in restoring the vision. So if time permits, at the end of the presentation we'll probably take a look at a couple of videos, and then the fourth section is conclusion, where we conclude the presentation by presenting an analysis of advantages and disadvantages. Visual prostheses, often referred to as bionic eye, these are mainly intended to restore functional vision in those suffering from either partial or total blindness. There are different types of implants which aid in restoring the vision. The first type is the cortical implants, where the stimulation is done to the visual cortex using a set of electrical impulses. And the second type is the optical nerve implant, where the optic nerve is stimulated externally. And the third type are the retinal implants, which are further divided into 2 categories. The first type is the epiretinal, and the second type is the subretinal, which is the topic of our interest. It is important to understand the anatomy of human eye in order to understand the way vision is perceived. So in the couple of slides which follow this one, we'll be talking about some brief aspects of human eye anatomy. Cornea is a clear dome-shaped window in the beginning of eye, which acts as a window for the light to pass in, and sclera is this white protective coat which protects the eye. Lens is a crystalline structure that focuses the light into the retina. It adjusts the shape for close and distance vision. And retina is the sensory tissue where most of the processing happens. So it converts the light energy into the electrical impulses which are used to communicate back to the brain. And these impulses are carried by the optic nerve, which transmits the visual information from retina to brain. Retina is further divided into 10 different layers. All these layers comprise of 3 different types of cells. The first one are the photoreceptor cells, and the second type are the neural cells, and the third type are the support cells. Analogically speaking, this can be thought of as a film inside a camera. Photoreceptors are further divided into 2 categories. The first category is rods, which function primarily in dim light and provide black and white vision. It is believed that there are about 120 million rods in the human retina. And the second type is the cones, which function best under illuminated conditions and provide color vision, and these are believed to be around 6 million. There are 4 types of neural cells. The first one is the bipolar cells, which connect the photoreceptors to the ganglion cells, and these are vertically oriented, and you can see from the figure that the rods and cones are connected to the ganglion cells using the bipolar cells. And the second type are the ganglion cells, which whose axons basically extend into forming the optic nerve. And the third type of cells are horizontal cells, which are horizontally oriented to and connected in between the bipolar cells. And the fourth type of cells are amacrine cells, which connect the bipolar and the ganglion cells with each other, and they function within the inner plexiform layer of the retina, where bipolar cells and retinal ganglion cells form synapses. Each type of amacrine cell connects with a particular type of bipolar cell and generally has a particular type of neurotransmitter. Support cells comprise of 3 different types of cells. The first type are the Mueller cells, the second type are the astrocytes, and the third type are the microglial cells. The main function of these support cells is mainly involved in maintenance of the retina. Now, having discussed about the anatomy of the human eye, we now talk about common diseases and disorders affecting the retina and thereby inhibiting the human vision. The first commonly known disease is age-related macular degeneration, which involves the degeneration of macula, which is the central region of the retina. This is a leading cause of blindness in the people of age groups above 65 years, and the degeneration of macula basically results in the loss of central vision. This is presented in 2 types, which is dry AMD and wet AMD. The second type of the disease which we are going to talk about is retinitis pigmentosa, which is a progressive eye disease that damages the rods and cone cells of the retina. This is known to be caused by genetic effects. It first attacks the rod cells, causing a progressive loss of peripheral and night vision, Eventually the cone cells are attacked and therefore the person gets blinded completely. So the pictures distinguish different types of vision. The first one is a normal vision, which is perceived by a normal human being, and the person with AMD vision perceives the same image in this fashion. The person with retinal pigmentation, retinitis pigmentosa, perceives the image this fashion. In order to overcome the deficiencies caused by these diseases, we use visual implants. It is believed that the electrical stimulation of the visual cortex makes a blind subject see a phosphene, which is nothing but a spot of light. Further, the spatial location of the phosphenes are influenced by the location of the electrical stimulus over the cortex. The current artificial vision techniques encash on these principles. Further, it is believed that the electrical stimulation of neutrons makes it an open voltage sensitive ion channels, thereby rushing inflow of sodium inside the cell membrane, which triggers an action potential, and thus it is transmitted to the next neuron. So the available implants in the market are broadly classified into two categories: the retinal and optical nerve implants, which are basically used for the diseases that result from outer retinal degeneration, examples being the AMD and RP diseases. The other type of implants are cortical implants, which assume that the entire visual pathway of the subject has been compromised. This is due to the whole thickness retinal diseases or optic nerve diseases or eye loss or central nervous diseases, etc. Subretinal implants. These were developed by the team led by Eberhard Renner, and these are used to substitute the lost photoreceptor cells in the retinitis pigmentosa disease, where the rods and cone cells are attacked. And it assumes that the most of the nervous tissue, which has the task of pre-processing information, is intact. It just means that the visual apparatus is functional, it's just lacking the input. And it replaces the natural optical stimulus with pulsed light-dependent electrical stimuli, resulting in the perception of the phosphenes. This is located subretinally behind the retina. The advantage of being located subretinally is that it exploits a full range of neuronal circuitry in the retina all along the way till the optic nerve. The electrical signal which is triggered by these subretinal implants are directly dependent on the intensity of the light which is incident on them. In this slide, we are going to talk about the architecture of the subretinal implant. The subretinal implant consists of a microphotodiode array, MPDA, which is of the dimensions 3 cross 3.1 mm, and each unit of this array consists of a microphotodiode, which can be seen in the expanded view here, and there is also a differential amplifier to amplify the incident light signal and then convert it into an electrical stimulus through a local stimulation electrode. And each individual unit corresponds to a pixels resolution. So there are total 1500 such units approximately. Actually there are 1520 units, which is around 38 cross 40 resolution, and each unit has the dimensions of 50 micrometers, not 20, 50 cross 50 micrometers, and separated by about 20 micrometers. And apart from these microphotodiode array, there is also an external electrodes, about 16 quadruple electrodes, which are used for testing the efficiency of the subretinal implant by electrical stimulation. So rather than relying on the light alone, the subjects can still perceive images by stimulations which are input through these electrodes. And this entire subretinal implant, the diodes in this implants are basically responsive to the light corresponding to the visible spectrum, which is around 400 to 700 nanometers. And yeah, as we have discussed before, the chip basically generates 38 cross 40 unique patterns. And the chip is said to consume very minimal power, which is about 10 milliwatts max, and the heat passed down to the retina by the electrical power generated from the chip remains below 0.5 K. Now, having discussed about the architecture of the subretinal implant, we now look at the performance of the subretinal implant. So these are the series of tests conducted by Dr. Renner's group. The test sequences include testing with the electrical stimuli on the DS test electrodes, and then followed by light evoked tests. Upon passing successfully, there were tests for recognition of stripe patterns and object recognition and visual acuity assessment. If all of these tests were passed, then more challenging tasks such as recognition of projected targets, were performed, and all of the tests were performed separately in two conditions, with the power source enabling the subretinal implant in on condition and off condition, and the results will be viewed in the following videos.
Letters, you know where they are, and then you order them according to what we present onto your direct simulation field. Okay. So we start with a letter. I put it here so everybody can see of us what we're presenting you now.
Now it's V.
And then comes the next one.
Okay. O.
Okay, and the next one.
L.
And the next one.
T. Tap something on the top of it.
Okay, so the word?
Volt.
Okay, we have only microwave, but
Right.
is nice. Okay, thank you. Great, you did it correctly.
So, okay, what I feel like seeing, I see a table right here.
Yeah.
I mean, plate.
Plate, yeah.
And then there's a knife here.
Yeah.
And this one looks different on the right, so I would say it's a spoon.
Mm-hmm.
And then there's a mug here.
So far we have looked into the architecture of the subretinal implants. We have also looked at the results when these implants are embedded into the subjects who are blind, and they were able to perform tasks better. Now we conclude this presentation by presenting an analysis on disadvantages and advantages. The advantages of using subretinal implants are: it has an edge over epiretinal implant because of its simpler design, and it requires minimal architecture space, and it allows for normal inner eye retinal processing by relying on the function of the remaining retinal layers. Also, because of the subretinal implant, subjects can use normal eye movements to shift their gaze. The disadvantages of using subretinal implants are the MPDA needs adequate amount of light incident on it to generate current. Hence, this might not be the case all the time. Hence, we need an external power supply to amplify the effect of incident light. Also, the compact nature of the subretinal space constrains the size of the implants. And because of the chip embedded onto the retina, the possibility of thermal damage is an issue because of the close proximity between the implant and the retina. And more importantly, the subretinal implant rely on the functionality of the remaining of the retinal layers. Hence, in the cases where the diseases are not pertaining to retina, rather where the entire visual system has been compromised, these might not be of good use. Thank you.